So the project we're working on is assigning functions to uncharacterized proteins. This is the team that I was working with. So we got me, Jim, Chris, Henry, Gordon, Ross, Alan, Gary, Alex, and Justin. And we had a lot of people floating in and out during the entire Codeathon, which was nice. The project goal is to predict a function of uncharacterized genes using machine learning. And we're going to predict it using various models or features. They span from whole nucleotide acid sequences to k-mers to codon usage to synteny or gene neighborhoods, as well as somebody suggesting using a fairly large pan-genome metadata vector. We'll get a little bit deeper into that right now. So the traditional machine learning would be something like XGBoost, random forest, and we're applying to those to nucleic acid k-mers, synteny gene neighborhood models, and a codon usage. Not all these models did get built. We were trying to build the matrices and get everything running. We did run into some hiccups here and there. With the large language models, we were trying to integrate GenSLMs and Hugging Face for fine-tuning, and hopefully fine-tune, not a Hugging Face, a GenSLMs model for predicting the gene function. Somebody had a suggestion of trying to use a fairly large pan-genome metadata vector. He wanted to combine features from 16 different sources of metadata and use some AI to merge all of these into predictions. The data set we're going to use, in order to calculate accuracy, we need some sort of ground truth. And an ideal data set would be characterized labels on uncharacterized genes. Unfortunately, this doesn't exist. So our data set consisted of characterized genes where we mapped a gene ID to a gene product string that was from BV-BRC. This gave us a total of over 8.8 million genome unique sequences, which were downsampled to about 250,000. And in total was about 5.5 thousand unique classes to train on. With respect to modeling, the traditional ML again was nucleic acid 7-mers, codon usage, synteny, and then we did try to do a GenSLMs model using full nucleic acid sequences. So with respect to the codon usage, so each person asynchronously went out and did their own thing in the group. We had multiple approaches. So Jim tried to do codon usage. He found a tool to compute codon usage, and he computed the codon usage on about 20,000 genomes. Unfortunately, we did not have enough time to actually train any models on this. Gordon, Ross, and Alan were doing synteny stuff. Gordon had had some codon previously to do some of this. So they computed synteny on roughly 9,000 genomes. They still need to compute a training matrix to actually train a model on this. On the plot to your right shows a UMAP plot showing basically distances between the gene neighborhoods and how they cluster together with respect to what kind of gene it actually is. The stuff in red are known genes. The stuff in black have no function on them. And you can kind of see there is a little bit of black underneath this red area here, but it does look like it is two complete clusters that we'll talk about more later on. One of the models that we did get running but not completed and not anywhere near where I'd want it to be were the 7-mers. These are the ones that I ended up building. And I computed 7-mers for all proteins and got a training matrix out. We began training models and ran into a lot of issues where computers crashed, we ran out of RAM, and at one point, the model just kept predicting the same function. So I had to work and try to find ways around this. The accuracies are a mixed bag. Some of them are fairly high, while others are just guessing. The macro F1 scores are shown to your right. 50% is guessing. Anything above that is better than guessing. So there's some signal in some areas, lacking signal in others. Note the model only had 50 samples from each of the actual functions, so it may not be enough data to actually train on. With respect to GenSLMs, Alex is still churning array, getting the Hugging Face integration. It is currently in progress. Hopefully by the end of the Codeathon, something is up there. He was able to generate a plot that basically took all the embeddings and put them into a 3-dimensional space so we can kind of see how things clustered out. The important thing with this plot is that the pinkish dots are uncharacterized or unknown gene function, while the red dots are hypotheticals. And you can see all the hypotheticals seem to cluster around the upper left-hand corner, while the unknown uncharacterized genes kind of just distribute throughout. We'll be talking about this a little bit more in just a bit as well. Chris was the one that had that entire pan-genome metadata vector, and he finished data scraping for 2 of those 16 data sources. Potential impact would be gene function. Can we do an ML-based annotation system? Does that work? Are the accuracies high enough? And as of right now, they absolutely are not. But with more data, we don't know. The other question is, can we actually apply this to uncharacterized genes, which is the main focus of this project? And one of the assumptions that had originally come in with this project was that the model can apply to uncharacterized genes, but the question is, do they really? And you can see with all the hypotheticals in the two plots that we did here, the hypotheticals tend to cluster in the upper chart with all the red in their own little corner of the world. And if we don't have enough proteins in that corner, then it's not going to apply properly to hypotheticals. And you can kind of see that sort of split in the lower plot as well. With what Alex did with the uncharacterized genes, we can kind of see that they do actually flow through the entire vector space. So you might actually be able to apply our models to those if they get accurate enough. There's still a lot of work to be done. As I said, we ran into a lot of compute issues, but we still need to finish training matrices to codon usage, synteny, and other stuff. Our traditional ML ran into a lot of issues. Part of it's due to the hacky code. This is a Codeathon, and we still need to tune model parameters, but also increase the dataset size. Hopefully, GenSLMs Hugging Face integration will also help out as well. That's all I've got.